Hello and welcome to the Map of the Month. My name is Harry Salt. I'm a neuroscientist by background and I do content creation in the life sciences space. Today I'm joined by Thomas. I was wondering, Thomas, if you could give us an introduction and a bit of your background.
Yeah, sure. So, hi, I'm Thomas Topilko. So I'm the co-founder and lead scientist at Vibraint, a Copenhagen-based startup building a platform to accelerate neuroscience research and drug discovery in general. So my background is at the interface of neuroscience, computational biology, and 3D imaging. I did most of my studies in France, in Paris, as you can probably hear from my accent. And my first introduction to tissue clearing that then became my scientific obsession came almost 10 years ago, like a decade ago, during a bachelor's internship in the laboratory of Alain Chedotal. And so I still clearly remember seeing the first sort of mouse brains and even human embryos that were made transparent in test tubes. And at the time, Alain's lab was probably one of the first in the world that have access to commercial light sheet microscopes. So it's like these specialized microscopes that enable actually the scanning of these samples, of cleared samples. And Alain was studying brain development and axon guidance, and seeing those data sets, I just remember, was mind-blowing, right? Because we come from, as neuroscientists, we just study thin slices of tissue, right? So just seeing intact whole organs that are transparent is just mind-blowing. And then I went on to do a master's in the US at Rockefeller University, so in the lab of Marc Tessier-Lavigne, and there I met someone called Nicolas Renier that turned out to be my future PhD mentor. And so Nicolas, he was applying tissue clearing techniques, again the same thing that I learned back then, more on mouse brain and studying how the mouse brain is not only built but functions. And after my master's, I joined his newly funded lab in Paris, and I did my PhD there, where of course I used some of these techniques to study more like fundamental questions. So I was actually focusing on parental behavior in mice, and more particularly nest building during pregnancy in pregnant mice. So we were like trying to figure out what are like the neural underpinnings of these switches of behavior that occur during pregnancy.
That's really interesting because it's a very automatic, instinctive response, isn't it? That kind of behavior.
Exactly. And so what we're going to talk about now, right? Like c-Fos mapping in general and this concept of activity mapping, this is sort of what we used during my PhD to understand what are the centers of the brain that could be responsible for driving, these changes. So there's very strong changes in behavior that occur during pregnancy that are obviously thought to be driven by hormonal changes in the animal. The idea was to try and use these techniques to understand what the the centers in the brain that could control these behavioral changes. And then in the last 5 years, I moved to Copenhagen, and I worked in two companies, so Gubra and Lundbeck. That's where I met Jakob, where I applied my expertise in 3D imaging, but this time in more industrial settings, both for contract research customers and more internal drug discovery programs. And now, about 8 months ago, we started together Vibraint. So our goal is to build on this experience and make advanced whole brain imaging and analysis more scalable and useful for neuroscience researchers and CNS drug discovery in general.
Well, I think that leads on quite nicely to talk about this map, based on your work at Vibraint. And I'll show a representation of how that looks now on the screen. But do you want to talk us through it and what we can see here and how this kind of fits into what you're doing now?
Yeah, so this map of the month shows the brain's response to a drug that I think barely needs an introduction at this point. So the drug is called Semaglutide and is better known through its brand names Ozempic and Wegovy. And I think it's fair to say that these drugs have completely transformed the treatment of type 2 diabetes and obesity. And at this point, it's almost impossible, to be fair, to open any newspaper without seeing it mentioned.
Indeed.
So the way the way semaglutide works, at its core, it mimics a hormone that is naturally produced by the gut called GLP-1 or glucagon-like peptide. And this hormone is released when you eat, and its job is basically to help coordinate the body's response to food intake. And so it stimulates the pancreas by releasing insulin, reduces glucagon, but more importantly also slows digestions and sends signals to appetite circuits in the brain that control food cravings. And so the problem is that in its natural form, that hormone is a terrible drug because it disappears very quickly. It's essentially broken down within minutes by an enzyme called DPP4. And so semaglutide was specifically engineered to overcome these issues. First, it resists the degradation by this enzyme that I just mentioned, but also binds to a protein in the blood called albumin, which gives it a much longer half-life. It makes once weekly dosing possible in humans. And what makes this map, I think, especially interesting is that, semaglutide is made in Denmark, but
I
it's
didn't know
not.
that.
Yeah, so it's made by a company called Novo Nordisk here in Denmark. But I think the key thing is that it not only acts on the pancreas, but also on the gut. And more importantly, it's very clear that it also works through the brain. So it engages circuits involved in appetite, satiety, interoception, so the sense of the body internal state, so to say, and nausea, which is a familiar side effect of many of the GLP-1 drugs. So it's important to realize that what you're looking at right now, this map, is not just a simple drug effect, but it's sort of like a window into how a metabolic signal, one that normally starts in the gut after you initiate a meal, becomes a brain-wide response.
And there's some quite clear bright spots here around different areas of the brain, as well as like a sort of a general glow of the c-Fos. So can you speak to a bit about some of these different areas and what you're seeing from this information here?
Yeah, exactly. So we can discuss c-Fos a bit in a bit, but the idea again that this map represents the general sort of reaction of the brain in response to the dosing of this drug, right? And so what you see, these areas in the brain, those are areas that are actually known to be involved in appetite regulation. There's quite a bit of research published on that subject. All these areas have a specific name that is usually attributed to an atlas, right? An area in an atlas. But what's really key, and we can dive in this further in a bit, is that anatomical boundaries from these atlases are, in a way, arbitrary, right? It's some scientist at some point in time that decided that an area starts here, ends here. Of course, it's based on cytoarchitecture and maybe some specific markers that are expressed by cells in the brain. But overall, the point being that these boundaries, similar to countries, right, in geography, are artificial most of the time.
Yeah.
And so what you see are sort of functional units in the brain, clumps of cells that react to the administration of the drug, but it doesn't necessarily mean that they actually behave as a region, right? It's more like a functional unit in the brain that responds to the drug.
Interesting. And what were you using this specifically? Can you say what this was being used to investigate specifically?
Sure. So I think, one of the key things when, of course, if you're a neuroscientist, you just want to understand how the brain works. But I think it's quite exciting in the recent years, I've gotten quite excited about using sort of drugs, and more importantly, small molecules, compounds that occur also naturally in nature, how these actually hack their way through the brain, right? How do they alter brain function? And can we learn anything from the way these drugs act on the brain to understand the brain itself, right? In this case, so semaglutide, again, it's a GLP-1 agonist. So of course, part of its action is driven by GLP-1 biology, right? So the molecule actually binds to cells in the brain that express the receptor for GLP-1. And this, of course, has a cascading effect, right? Where you not only activate the cells that are directly responsive to the drug, but also all the cells that connect directly to these cells as a side effect, right? And so what this map shows, and I mentioned briefly before, is it's important to realize that what you're seeing is not only the direct effect of the drug. This can teach you a lot about the mode of action of a compound, right? Which is something absolutely key for anyone making a drug. But more importantly, it can tell you more about, what are potential side effects of the compound. Some of the signatures could potentially mean side effect, but also, some downstream circuitry, areas in the brain that are connected to the primary cells that are recruited by the drug, but that are probably fundamental in the downstream effect of the compound.
Thank you. That's great. Shall we move on to talk a bit about c-Fos then? Because that is integral to creating these maps here. So for anyone coming into this fresh, what is c-Fos and why has it become such a useful marker in neuroscience?
Yes.
And why do you love it so much?
Sure. Yes, the c-Fos, to be honest, is a weird one because I don't know if you know, but its origin story is not in neuroscience at all. I believe it was like in the 60s or 70s where researchers studying a mouse virus that caused a specific type of bone cancer called the FBJ, I believe FBJ osteosarcoma. So the cancer-driving gene in that virus was called the v-Fos for viral, and FOS was basically an acronym for FBJ osteosarcoma. But later, researchers realized that this viral gene had not come out of nowhere. It had been derived from a gene that existed in cells. And the normal sort of version of the gene was creatively named the c-Fos for cellular FOS, right? At first, c-Fos was studied mainly as a proto-oncogene, so a normal gene that when misregulated basically triggered cancer. But then I believe in the mid-80s, the story took a little bit of an unexpected turn because scientists discovered that c-Fos could be switched on within minutes after cells received a strong signal. That made, they basically called it from then on an immediate early gene or a rapid response gene, so to say. And it turned out that actually neuroscientists realized that it's extremely useful because neurons do this too. And after certain neurons become active, they switch on c-Fos, and the c-Fos protein appears in the nucleus of the neuron, a little bit like a flag saying, I was active not that long ago. So it's not a direct live feed of brain function, but more like a snapshot, right? It doesn't capture every neuron that fired, and it does not tell you exactly the sequence of events, but it gives you a powerful sort of snapshot of which cells were recently active at a given time. And that is why I think c-Fos became such a powerful workhorse tool in neuroscience. It has been used in thousands of studies to map brain activity, to understand the many functions like, stress, pain, hunger, sleep, and so on. And now with a whole brain 3D imaging specifically, so the technologies that we use, we can look at c-Fos across the entire brain, if not the entire central nervous system in one shot, right? Whereas before, this tool was equally powerful, but you had to basically slice up the brain and look into specific sub areas of the brain in a more biased way, if that makes sense.
Yes, absolutely. And of course, here it's being used, so c-Fos is the marker, but it's been used alongside semaglutide. So how well does it work as a readout for pharmacological effects more broadly? And how does it tell?
Yeah, so
in ways that other methods can't.
Of course. The c-Fos is, I think it's super useful, right? So for some of the reasons that I just told you about, because it basically lets us ask a simple question. It's, after a stimulus, which neurons were recently recruited by the stimulus. And that stimulus can be many things. It can be a drug, it can be a stressful event, it can be pain, it can be sensory input. By staining for c-Fos, you basically get sort of a spatial snapshot of recent brain activity. And when we combine it with tissue clearing and light sheet imaging, that snapshot can cover the entire brain, as I mentioned before. Its biggest strength is discovery, in my opinion. So c-Fos can reveal, the regions you expect to see, of course, but more importantly, it can allow you to find ones that you did not expect. And I think this is specifically true for the brain, where it's a very complicated heterogeneous structure, and the likelihood of you hitting the right area just by chance is, realistically quite low. So especially if you're fishing for subtle effects or rare events, these techniques are extremely powerful. But at the same time, I think that, like any powerful tool, it has its clear limitations, right? So it's definitely not a live recording of brain activity. It's delayed, meaning that usually you need to wait anywhere from an hour to 2 hours after a stimulus to actually see a strong protein signal. And it's more biased towards a strong neuronal activity. Importantly, not every neuron expresses c-Fos, so that's really important to remember. A lack of c-Fos does not necessarily mean lack of activity, but perhaps, I think most importantly, c-Fos does not tell us whether those neurons caused the behavior, right? So the only thing it tells you is whether that the neurons were recruited around the time of the stimulus. The causal implication, you need follow-up experiments to actually prove that. I think c-Fos, just to conclude on that, like c-Fos, I think is best seen for me as a brain-wide molecular snapshot, right? Extremely powerful for finding candidate circuits, but something that needs a clear follow-up with causal tools like optogenetics, chemogenetics, more standard things like electrophysiology, 2D, calcium imaging, and so on. So it's really a discovery tool that helps you have the big picture, but you really need to, it doesn't completely remove the use of more traditional neuroscience tools as follow-up to this.
Yeah, and there's clearly a lot of intricacy to even just the c-Fos itself with regards to the timing and how you're administering it there. But there's all of the other components of this workflow as well. So can you talk through how you actually make one of these maps from the tissue to the imaging itself?
Yeah, sure. So, we've been doing this for over a decade now. And these techniques, the fascinating thing is that tissue clearing, so the process of making tissue transparent, as well as light sheet imaging, which is this fancy way of basically generating a thin sheet of light to actually scan the cleared tissue using essentially optical sectioning. These two methods were created in the early 1900s. So it's nothing new. The reason why it was actually never mainstream is, I think, because of some of the other key components that were unavailable at the time to make these techniques shine. And the two components that I'm thinking about are first data collection, so more IT infrastructure. How do you store these data sets? And just to give you an idea, so if you scan a whole mouse brain with decent resolution, these are hundreds of gigabytes, if not terabytes of data. So of course, in the 1900s, storing terabytes of data was probably not an option. That's problem number one.
And just
sure.
And just quickly on that as well, with the amount of information, like how many slices are we talking here? Like how thin are these slices, and how many images roughly are you taking for that mouse brain? Just to put it into even more perspective there.
Sure. I think that a good comparison, I like this analogy that I read somewhere that, in classical 2D histology, if you make a thin slice of tissue, right? And this is typically how any neuroscientist study brain function. You aim for anything from a couple microns to maybe 10 microns thick, right? But if you, this only represents like maybe 0.1% of the tissue block, right? So I think a good analogy is like you're trying to understand a novel by reading a single sentence in the book, right? It's not really like, sure, it's useful if you know exactly where to look at, but in reality, especially if you think of the brain as a very heterogeneous structure where things are very different depending on if you even move a millimeter or 100 microns away from a specific area, suddenly all of this becomes very problematic, right? And so to give you an idea, yeah, I don't know. So usually we scan at a couple micron isotropic. So when I say isotropic, it means it's the resolution across all 3 dimensions in space. So maybe 2-3 microns in each direction. So it's thousands of images across the entire 3D volume. And then so you acquire these images sequentially, and you sort of reconstruct it volumetrically after the fact once you image the sample in 3D. And so just to go back to your initial question, right, of like how do we actually process the tissue and so on. So the way this works is from the business side of things is we receive tissue, right? Here in Copenhagen, usually just people ship us brain tissue, or we do the experiment in-house. We usually stain the tissue. So this is either using antibodies or using in situ hybridization, so RNA molecules, or maybe, the animal was injected with a fluorescent drug prior to the termination of the experiment. So we stain for something in the tissue. Then we collect the tissue and we clear it. And this is a chemical process that sounds very fancy, but in reality, it's extremely simple. The idea being that the brain is basically a mixture of fat, water, and proteins, and what we want to keep is proteins. So we remove fat, we delipidate the tissue, and we use a pretty harsh chemistry to do so, organic solvents, and then we actually dehydrate the tissue. So we remove water, and everything that remains is a mesh of proteins, right? But funnily enough, this is enough to actually keep the tissue like perfect integrity almost. And then we use again a pretty simple trick that maybe anyone that went to high school knows is something called optical matching, so refractive index matching, where I think the typical experiment you do in high school is you take some oil, right, and then you dip an empty glass in the oil. So if the glass is not filled with oil, you obviously see the glass, but the moment oil enters in the glass, it completely vanishes, right? Because the optical properties of the glass and the oil are similar, so the light doesn't scatter when it travels through the container. And it's essentially the same principle here, where you take that mesh of proteins, the brain, whatever remains of it, you dip it in a liquid that matches the optical properties of that mesh of protein, and it essentially vanishes, right? But the cool thing is that it makes it permissive to light, so light can easily go through the tissue undisturbed, so to say, or with minimal disturbance. But at the same time, we preserve fluorescence, right? So the staining that we did up front is preserved. So if you now shine light and you put your tissue in this special microscope called the light sheet microscope, you can actually visualize and collect data in 3D of the fluorescent signal volumetrically, right? Yeah, this is more or less how it works in a nutshell.
Excellent. Thank you very much, Thomas. And just to wrap that up, can we talk a bit about the antibodies that are involved in this as well and the role they play?
Yeah, so the staining in 3D is surprisingly one of the biggest challenges still, because we rely on a chemical process called passive diffusion. So you basically drop in your molecule, right? Usually an antibody that you want to stain a target protein with, and then you just hope for the best, basically. So you let the antibody diffuse in the liquid. You usually optimize the conditions so that the antibody penetrates or diffuses as fast as possible. One easy trick is just increasing temperature, right? But the idea is really quite simple. You just drop your antibody and you wait for the antibody to diffuse deep into the tissue that you're trying to stain. But the one key thing that I wanted to mention here is that the quality of the antibody that you're using here is absolutely critical. And my honest rule would be that once you find one that works, just stick to it. Never change again. Because that's especially true for c-Fos. If we want to compare brain-wide activity patterns across experiments, animals, treatments, and so on, the staining need to be consistent as possible. And small differences in antibody performance, protocol, or even antibody lot can create differences that look like biological signal, but to be fair, are most likely artifactual. So for our c-Fos staining, we use an antibody from cell signaling currently that we're very happy with. But again, my rule of thumb is you just need to find something that is reliable, and more importantly, something that the community, the neuroscience community, sort of uses.
On.
Correct. Exactly. As long as you have these two, you're good to go. That would be my recommendation.
And to move on from that as well, then to the taking the images and turning them into a whole brain map. One of the things you mentioned earlier was about the atlas and how it's kind of these arbitrary definitions of how you're defining these regions and characterizing, where they sit anatomically. So what is the process of taking those images, aligning them, registering them, and quantifying the data?
Yeah, sure. So as I mentioned, right, like tissue clearing and light sheet imaging is fairly old as a technique, and I would say that the last decade, right, if you look 10 years back, most people use these techniques to generate beautiful images. So you just make these beautiful images, and usually people put it as like a supplementary figure in a paper or a movie, but they didn't really use these techniques in a quantitative manner. So really the critical part right now is to turn these beautiful 3D complex images, intricate images, into something that is quantifiable and comparable across animals. And this is something that I think has been, there's many labs now working on this in academia. And I would say over the last 5 years, quite a few tools were developed to really quantify things in 3D. And when I say quantify, it could be anything. It could be nuclei, it could be cells, it could be blood vessels, it could be axons. So different types of objects in a cleared and imaged 3D brain. But more importantly, to then not only segment these things in 3D, but then to also transform them into quantitative metrics that you can run statistical tests on and, do proper statistics using these type of data sets.
Because that is a native challenge with imaging as well as just being able to quantify it. And obviously when you're working with such fidelity and the methodology is so important to the outcome of the image, and being able to replicate that, I guess, across different studies to build up data to support certain things is really important.
Yeah, and that's where it's a bit tricky, right? Because to be honest, we've been working with that technology, again, as I said, over a decade. And for us, some of these things feel obvious. But I think you quickly realize that to really implement that technology in a lab setting or a pharma setting or biotech or whatever, you really you usually need a small dedicated team of people, right? Each one with their own sort of specialty, like maybe one person that is really good at the clearing part, right? So histology, one person that's very good at imaging, one person that's good at the analysis part. It's quite tricky to find a person that has many of these skills combined in one person. And this is really limiting, right? When you want to try and implement these techniques, because you have to find sort of specialists in all these different parts of the pipeline to make it usable. And as you said, I think one critical thing is that any step of the pipeline is really crucial. Like if the garbage in, garbage out is a good analogy here, right? So if you don't process the tissue properly, then you might be the best analyst in the world. There's not much you can do. So every component of that chain is very, very important here.
Yeah, and looping back to where you were talking about this process of the registration, how does that actually work in reality, aligning these components?
Sure. So Actually, the way alignment works for 3D data is we use something that is slightly counterintuitive, I would say. So we take advantage of something that is usually like a microscopist nightmare. It's called the tissue autofluorescence. So autofluorescence is like the natural fluorescence of the tissue, and it comes from molecules such as lipofuscin, flavin, collagen, and other endogenous components that are naturally present in the tissue. And usually this is a nuisance, right? Because it creates background in the tissue and it actually interferes with your specific fluorescence staining that you want to record. But for registration, it's extremely useful because we usually acquire a dedicated autofluorescence channel and use it almost like an anatomical fingerprint of the brain, if that makes sense. So this channel gives us enough structural information to align each sample to a common reference brain, and the reference brain is just an average of hundreds of brains that were collected previously, right? So mathematically, I don't want to go into too much detail here, but the registration algorithm looks at the 3D pattern of intensity, the gradients of gray values in space, and it tries to basically align and deform your sample onto that reference. And once the alignment is successful, you then apply the same transformation to the coordinates of detected objects, whatever the objects you're interested in segmenting is, right? And the result of that is that every detected cell or object is now placed into that common anatomical coordinate system, right? So, and that is really the key step. It means that all the animals, all the treatment groups, all the experiments now live in the same reference common space. From there, you can not only quantify the density of a certain object per brain region, but also create, more importantly, group averages, compare drug effects, and generate brain-wide maps, like the one that we're looking at right now.
Yeah. And you've already touched on many challenges, but what would you say is some of the trickiest parts of this kind of experiment? Like, where do things tend to go wrong, and where do you have to be really careful?
Yeah, so I think it's actually one of the key things that we are focusing on now as Vibraint, the company, is one of the inspiration came, I think, from my own frustration looking at these data sets. Again, I think going from a beautiful image to something that is quantifiable to doing this high throughput is already quite good, right? But I was always frustrated by the fact that the only information that you get from that complex three-dimensional image, that the map that we're looking at, is only the specific location of an effect, right? That is the key information that we can extract from these maps, where something happens. But the idea is that we want to go much deeper than that. We want to understand what the cells underlying these effects, these brain-wide patterns could be, and so on and so forth, right? Like a much deeper understanding into the actual components underlying those patterns, intricate patterns of activity. And so a lot of the tools that we're creating at Vibraint now is trying to take different types of data, what we call multimodal data, but it's basically other modalities of data. So it could be spatial transcriptomics, it could be whole brain connectivity data, it could be functional MRI, for instance. But the idea being that you bring in these other types of data sets in the same framework, and you can actually now predictively try and understand or infer, okay, you have an activity pattern in that small spot in the brain. Is how many different population of neurons live in that area? Let's say 3. Then, there's only 3 possibilities now instead of potentially dozens. So in a way, you go from this like very speculative way of looking into these things and more data-driven, where you still have to run a follow-up experiment to understand what is actually going on, but you make these decisions in a much more predictive, data-driven way than before. I hope that makes sense.
Yeah, no, it absolutely does. And Jakob mentioned as well about the CNS Voyager platform or program that you're working on. I was wondering if you could shed some light on that and where that fits into this bigger picture.
Yeah, sure. So I think I've always dreamed of having access to such a platform. So I'm almost like the first, hopefully the first big fan and customer of our own tool, which is also dangerous because I feel like I would be the kind of person that would just sit hours and just stare at, brain patterns. But in any case, I think the future is to move from these, as I mentioned before, these beautiful, just qualitative brain maps into something that is more interpretable, in my opinion. So whole brain activity already gives us this powerful overview of how the brain responds to a drug, behavior, or disease state. But the next step, I think, is to connect those maps to other layers of biology. And that means asking, questions like, as I said, which cell types are active? It's not only where do things occur, but what are the actual cells that respond to the drug in that question? Which receptors are expressed in those regions? Which circuits perhaps connect between the primary responding cells and the rest of the brain? Is the pattern specific to one drug or shared across multiple classes of compounds? Does this activity pattern predict efficacy, maybe side effects or disease progression? And this is really where I think the CNS Voyager platform becomes important, because the idea is to use these whole brain maps not only on isolated images, but part of a larger navigable framework, right? Where you combine activity, anatomy, cell types, receptors, connectivity, all these very different dense type of data sets in one sort of unified framework, right?
So it's more like an encyclopedia than a map in a way.
Exactly.
So you're having more layers of information on top of the images that you'd usually have.
Yeah, and to be honest, even as a scientist, like my dream would be to have access to almost like a Google Maps of neuroscience, right? Where you go, you log into that virtual platform, and
Yeah.
you maybe bring in your own data set, and you have access to these multitude of publicly available data sets that people share, that we generate, also internally, to try and better understand what is actually going on in your own data, right? Generated in your lab or in your company. And so for drugs like semaglutide, I think that means that you can start asking more mechanistic questions also. Which activated regions are likely direct GLP-1 receptor targets? So more mode of action type of questions. Which are the downstream network effects, perhaps? Which parts of the response are related to maybe appetite suppression, and which might be linked to nausea or aversion, right? So more side effect type of activity, for instance. But more broadly, I think this could help neuroscientists and drug discovery in the neuroscience space more generally move from this like single region obsession way of thinking, right, to system level interpretation. And so, of course, as a neuroscientist myself, is very appealing.
It's a really exciting. I mean, for anyone who's interested in science, I think that is an extremely exciting development to look forward to. And I guess the other side of that as well is the advancements in the hardware or technology itself, because that's software platform based development. But what about the sort of hardware advancements for whole brain mapping? Where do you see that developing in the coming years that might unlock more for these kind of platforms?
Yeah, so there's different parts of this. So I think if you speak of hardware, I think one part of this question is it's also about data, like everything we discussed before, right? Like the IT part of it. So how do we store larger amounts of data? What is the resolution at which we even need to store that data to extract meaningful information from? All these questions where I think it's not really solved problems. It's actually things that we're working on as we go. But it's of course an issue, right? Especially in the current sort of also geopolitical situation where the cost of RAM and GPUs is exploding. This is clearly a thing that we're also considering. as a company, right? So because as I mentioned, these data sets are not of trivial size. It's hundreds of gigabytes to terabytes, and even nowadays, like some people that we collaborate with, even in the petabyte range, right? So how on earth do you store this? But more importantly, how do you actually collect also all the metadata that's relevant for describing these data sets long term, make this data reusable, like integrate all these data sets together. And these are challenges that we work on every day. So it's I wish I had an answer. But yeah.
I think that's an exciting conversation to perhaps have another time to dig into that a bit further, because I feel like we could probably talk about the hardware for quite a while as well. But thank you very much for sharing your wisdom and insight onto regarding c-Fos, semaglutide, and what you guys are doing at Vibraint. It's great talking to you.
Yeah, thanks.